Welcome to science class. Today we are going to continue learning about genetic evolution by taking a closer look at how natural selection changes populations. In the last video, we focused mainly on sources of genetic variation and how that variation affects the population. But we were taking more of a snapshot view. In this video, we will be learning how genetic variation affects the population over time, not just how it accounts for the population as we see it currently. Let's get started. I read this book a few months ago, The Chemical Age by Frank von Hippel. A huge portion of the book is dedicated to the discovery and implementation of insecticides in times of war and times of peace. Insecticides were hailed as one of the most important scientific achievements in history for their ability to allow people to grow more food and to eradicate diseases transferred by vector hosts like fleas, mosquitoes, and lice. But it didn't take long for there to be a big problem. Say the insecticide kills 99% of all bugs because 1% of the population is resistant to the insecticide. We use the phrase breed like rabbits, but insects make rabbit breeding look like elephant breeding. So, in no time at all, the insect population is gigantic again. What percentage of the population is still resistant to the insecticide? If evolution wasn't real, then only 1% of the population would be resistant. But I'm sure you already know that's not how it works. Some of the insects got lucky and weren't exposed to the poison the first go around, but they won't be so lucky next time. But all of the descendants, or at least the majority of them, of the 1% who were resistant still are resistant. So next time, maybe 2% of the population has resistance, then 5%, then 10, then 50. Time and time again, farmers and chemical companies were finding out that one specific brand or formula of insecticide stops working after a few years. This is the definition of natural selection: the differential survival and reproduction of individuals with beneficial characteristics. And those characteristics come as a result of genetic differences. If we could sequence the genomes of these insects and find the allele for the gene that makes them resistant, we would see that allele gradually become more and more common over time. Let's look once again at single gene and polygenic traits. Single gene traits may produce distinct phenotypic differences. The mutation that causes peppered moths to be black as opposed to the usually more common white is a single gene mutation. If you remember from last time, this means that peppered moths will come in only two varieties. Their pattern of speckles will not always be the same, but their underlying pigmentation comes in just two varieties: this specific shade of dark or this specific shade of light. If the pigmentation trait was polygenic, then we should expect to see a range of pigmentations in these moths. If the trees are unpolluted, you would expect to see mostly white moths, and each pigment that was darker would become gradually less common. But again, that's not the case because it's a single gene trait. Polygenic traits are more fun to look at in populations. With single gene traits, they either fit into the environment or they don't. White moths are camouflaged and dark moths are not. But if camouflage is polygenic, then there are a range of phenotypes that have increasingly good or poor fitness, which again is why we get the normal distribution. But now let's see how that normal distribution changes over time. The first type of selection I want to discuss is stabilizing selection. While it is true that environments change, organisms evolve, and species go extinct, those things typically happen on very long timescales. The general rule in the short term with evolution is, if it ain't broke, don't fix it. It is beneficial for an offspring to resemble its parents because their parents survived to the point of reproduction, hence they continued their species. That's what you call winning the game of biology. And stabilizing selection is when natural selection favors the average. Take a look at these two bell curves. The range, or diversity we could say, of one is very great, while the other is more homogeneous, but they both have the same average. In stabilizing selection, what you see over time is the population begins to become more homogeneous. Let me remind you that we are only discussing a single trait, controlled by multiple genes. This doesn't mean that all the organisms become more similar across all traits. It just means that one particular trait is favored to be standardized, rather than coming in a wide variety. So here's a hypothetical example. Let's say we have an island, and we introduce a species of birds to the island. It turns out that these birds eat only one kind of seed. It wouldn't make sense for the birds to have beaks that come in a wide variety when each beak is used for only one purpose. So in this population of birds, we would see their beak geometry converge to a more narrow range. The next example of polygenic changes in fitness is known as directional selection. This is one of the driving forces behind speciation, or the evolution of new species. As I stated before, environments change, and when that happens, changes in pre-existing traits are required to prevent going extinct. Evolution is gradual, so an organism does not typically produce offspring with completely new traits, because that would require so many simultaneous and spontaneous genetic changes that it's statistically improbable. There's also the fact that evolution does not have foresight. Each trait has to be tested in the struggle for existence, so having rapidly changing DNA is not beneficial. Although there are what are called macromutations. Here is a frog with eyes inside of its mouth. Now that obviously isn't beneficial, but we can easily imagine a scenario where some other sort of dramatic change is beneficial. Okay, sorry, let's get back to directional selection. Let's plot the original phenotypic range of the population. If the environment changes in some way, then the average is no longer adequate. The species will have to modify some pre-existing trait in order to have greater fitness. Let's use our bird example again. Our birds become adapted to eat only one kind of seed on the island. Let's imagine that an invasive bird species moves in. This bird species eats the same seeds, but is more aggressive and has more offspring every reproductive cycle, so they have a big competitive advantage over our native bird species. If the birds want to continue surviving, they need to occupy a new niche. The seeds they used to eat are now unavailable, so out of necessity they start eating different foods. Their beaks work okay, but the birds with slightly smaller and more narrow beaks eat faster. Now they are competing with each other. The birds who can gather resources faster have better survival and reproductive potential, and this drives the population to change. We would see the normal distribution shift generation after generation. This exact same scenario could play out within the species itself. Let's imagine that the invasive species never arrives. The native bird population grows so large that soon there isn't quite enough food to go around. In response, some of the birds are forced by their own species to try and forage for new foods. This subset of the population then begins to evolve by directional selection, while the rest of the population stays in stabilizing selection. At first, this isn't a big deal, but after only a few years or decades, this can result in full-blown speciation. The two groups of birds that were once part of the same population become subspecies. Theoretically, they could still interbreed, but their morphology and behavior changes so much that they never do. I first presented this argument in lecture 33, but this kind of change has actually been documented in the wild. A famous example was the study of finches on the island of Daphne Major by Peter and Rosemary Grant. I'll let you look into that on your own if you want to. The final example of polygenic changes in fitness is known as disruptive selection. In this case, the species diverges in different directions because of selective pressures. This is similar to the example I just gave, where one subset of the population moves into a new niche and the other subset remains, but in this case, neither subset remains in stabilizing selection. So on our theoretical bird island, here's how this could happen. Our original bird population is in stabilizing selection, and they eat only one kind of seed. Then all of a sudden, the tree that produces those seeds goes extinct due to a blight, a fungal parasite that infects plants. This forces all the birds to have to occupy a new niche. The birds explore all their options, but they find that there are two different food sources that work well for them. One food source is found in another tree, and the other source is found on the ground. The birds that forage for food on the ground don't lose the ability to fly or anything that dramatic, but their behavior and morphology changes enough over several generations, and once again, we end up with two distinct subspecies derived from a common population. With each of these different graphs, we were tracking the phenotypes of traits. Genotypes are responsible for differences in phenotypes. So we could have studied how the alleles within the population shifted over time as well, but that's a completely different kind of analysis. It is, however, the subject of the next video, where we will investigate what causes the gene pool in two different populations of the same species to change over time. Thanks for watching.